Welcome, everybody. I think we'll go ahead and start. I gave a few extra minutes in case there were some Berkeley people that need a little time to get up here. It's my great privilege to be able to introduce our distinguished speaker today. Lisa Overius is here on sabbatical, actually, for about half a year. So we're taking advantage of her being here to give a seminar today. I've known Lisa for many years. We've had the similar communities during my time in Scandinavia. We got to know each other quite well. And Lisa is one of the first persons to have really worked with DNA in soil and to use methods to understand the extent of microbial diversity in soil environments. And that was in the 80s that you were working on that. But this is still a very much a hot topic, as we'll see from Lisa's talk today. A little bit more about Lisa's background. She's currently a professor in geomicrobiology at the Department of Biology and the Center for Geobiology at the University of Bergen. And she's also currently Dean of Research at the Faculty of Mathematics and Natural Sciences at the university. In addition, she is coordinating an EU project involving 19 participants from different countries, 19 different countries in Europe, that has the focus of developing methods to analyze complex data sets, such as what we work with. So that would be very advantageous for us to learn about that as well. In addition, she's on the committee for the U.S. National Center for Ecology and Analysis of Systems, so she's on their advisory committee. And by kind of coincidence, she found out that she happens to be here at a good time because they're starting the Petter Sæther Center in Berkeley, which is a collaboration between Norway and the U.S. To foster scientific collaborations. And so Lisa's also involved with that center development while she's here. And so I'd like to introduce Lisa to give her talk. Thank you.
Thank you, Janet. Thank you for inviting me to give this seminar and for being here in Berkeley. So I'm going to talk a little bit of what I'm doing here and also what I've been working on for the last couple of years. I titled my talk today, The Vanishing North. I actually borrowed the title from The Economist this summer. There was a big report on the vanishing of the Arctic and also the consequence for the community in the trade and economy. And also I could see that the science part of this was actually a quite minor part, but it was also the effect of what this climate change have on the whole community. So I'm here as a Fulbright stipend. I've been so lucky to receive something that is called the Fulbright Arctic Chair stipend. You probably all know the Fulbright Foundation, but this is the foreign minister of Norway that really want to focus on Arctic science from Norwegian scientists. And want to promote collaboration between Norwegians and U.S. scientists. So it was kind of, when I applied for this, it was kind of strange to know that I should work on Arctic topics, but it was perfectly okay to stay in California and to do this research. It's also people from U.S. that can get this scholarship, and they have to be in Norway and preferably at the UNIS, which is a university center that we have in the Arctic. And part of my this scholarship also tells me to promote the collaboration between Norway, US, and also especially in the university center that we have in the Arctic. So this shows that we have a university center in Svalbard, which is the world's northernmost institution for higher education. It's located 78% north. It's owned by the mainland universities in Norway, but it's driven and run from the Longyearbyen. It's It's open for international students. We have 50% Norwegian students and 50% international students. It started in '93, and it was a big political issue whether we should start with having teaching and education in the Arctic. And I have been teaching Arctic microbiology since 2001. And I'm also in the board for this Arctic University. After having started to spend time in the Arctic, I've been very excited about the environment, and it's also very fascinating to do research in this environment. This university center has a really good infrastructure, very good facilities, they do lots of experimental outdoor designs and doing very high quality science. So it's become a very popular place for scientists, but also for students. So this picture in the corner shows my student, 3 o'clock in the night, doing lots of field work. The sun is going down into the ocean, and they're making the UNIS logo while it's going down. So this is get really close to the environment and with the students when you are doing these classes in UNIS. So I hope that you will tell your students about this center and that you will try to come and use the facility and use the knowledge that is up there. So why is Arctic important, and why should we study in the Arctic? It's the most rapid warming region on the planet. It has constantly very low temperature. It has extreme seasonality, which is very striking. But yet it's a really biological active environment. And we also know that nutrients are turned over and pollutants are degraded in this cold environment. If we look at this figure showing the observed global temperature on the planet since 1850, you can see the blue curve is showing the temperature measured from monitoring stations around the globe. And then you can see that it's quite steep increase in temperature from 1910 to 1930. It's a plateau around 50, 60, and then again it's a quite steep increase in temperature. The red curve is from more modern satellites, but you can see that the conventional temperature measurements are coincident or correlate very well with the satellites. So there are natural fluctuations like El Nino and La Nina, and also some volcanic eruptions, but these are all into these models, and we can see that, or these data, but we can see that it's a really steep increase in the temperature. If we look into an animation of this, the temperature change from 1880, we can see that there are quite random, or the changes, natural variation are quite big. You can see the blue areas are when it's colder, when it's getting warmer, it's more into the red. So basically a lot of high variation, but when it comes to present time, it's we can see that it's getting more and more warm, and we can see that the warm is especially present around the poles. So around 70, 80 was pretty cold, and then it's starting also to warm up in around 19, and then we can see really increase in temperature. This animation stops at 2008, but also if we look at 2009, 10, and 11, we can see really dramatic changes in temperature. So this is the temperature changing. You can see also the Arctic is experiencing very high change in temperature. If we look into the CO2 concentrations, the first measurement started to be monitored around 1958, and this was monitored from CO2 in the air from Mauna Loa in Hawaii. And we can also see from 1958 they have a really steep increase of CO2 in ppm from 320 to 380 ppm. So it was more CO2 in the air today over the last 1 million years, and you can see the increase started after the industrial time, and the increase mainly is due to burning of coal, oil, and gas. So natural variation with volcanic eruption has also occurred, but it's really a minor input into the CO2 in the atmosphere. So now we have started to have a much broader measure, and it's an international global network of monitoring stations to monitor CO2. And you can see North America have lots of stations. They're more scattered in Europe. We also have a monitoring station up at Svalbard. But this is from 1958, when we want to go back in time to see how was the concentration of CO2 if we go back in time. This shows an ice drilling core from Antarctica, where it can go 400,000 years back in time. It's also a drilling project going on now where they take an 800 years back in time for the ice core. And there's also an ice core taken from Greenland that is used for this analysis. And what you can use these ice cores for, you can see that there are distinct layers in time. And also from this core you can do melt it and measure the stable isotope of hydrogen and oxygen, and that will give a proxy for the temperature back in time. And also if you do a gas analysis on the bubbles in the ice, you can measure the actual CO2 volumes. So this shows animation where this blue shows the temperature based on the hydrogen and oxygen values in the ice core back 400,000 years. And then this red curve shows the CO2 level in this core. And you can see that it really matched very well with the temperature. So the CO2 level and the temperature is pretty much the same or following. So again, natural fluctuations, but quite same numbers. So this is showing back until 1850, again the pre-industrial era. But if we go further and measure the CO2 level today from 2012, doesn't work. Okay, we can see that it's a really, really steep increase in the CO2 level in the atmosphere today. So up in 391.7 ppm. So this is the values that we have, we know are in the atmosphere today. So how do these influence on the Arctic environment? This shows a map from the pole. Here is Norway, where I'm from. This is Svalbard. You see the red line here is where the ice cap used to be from 1979 till 2000, and the white is where it was in the summer or September 2011. So it's been a really decrease in the ice in the pole area, which of course influence on the organisms living there, on the communities, people living around. But there's also a big issue whether these are opening up the North Sea routes and also the Northwest Passage. If this is going to be open and we can have ship traffic and things like that, which was the economists, was kind of how they were looking at this story. So as a biologist, we are kind of interested in the heterogeneous distribution of diversity. That's been have been triggering biologists for a long time. And also very striking that we have this high diversity if you go into the equator, the tropics, and that we have really low diversity if we come to the Arctic and Antarctic. Also, it's quite fascinating to see why we have different organisms at the different poles. Why do we have polar bears in the Arctic and penguins in the Antarctic, and why it's not vice versa? This is not going to be any talk about polar bears whatsoever. But I just have to mention, it's quite funny. I have been doing fieldwork in the Arctic for a long time, carrying the rifle in case we see bears. It never happened. And then after 2 weeks in Berkeley, I got this sign with a wildcat, mountain lion. Be careful. I was like, oh, do I need a rifle? No. Okay, so it's quite. Ecologists have been studying these for a long time to see, to understand why are they different in distribution and in patterns. And we that are looking at microbes, we have a little bit different story, different mechanisms that regulate this. This shows a sample from a soil in the Arctic. And we know that this is a gram of soil. We know that it's 10 to 21st stars in the universe. In this gram of soil we have, or in the soil at the planet, we have more than 10 to the 30 individual microorganisms that we at least know of today. It might be more we haven't seen. So exploring microbial diversity in any soil environment is more becoming like exploring the outer space with soil, the kind of final frontier. And also we know that this is an ancient group of organisms that have evolved for 3.5 billion years. So if we're interested in both the distribution, the abundance, and the evolution, this is really important to get to know more about these organisms. If we look at this diversity in the soil and look at if you calculate it into amount of carbon, we know that we have 5.5 from 10 to the 14 kilograms of carbon of microbes, and then everything else is just a small fraction of this. So this is when we also know that the microbes are the driver for the biogeochemical cycles, really need to get to know more about these systems. If we look into the soil in the Arctic, we know that the permafrost, which is the permanently frozen soil, represents about 37% of the northern hemisphere. So this is soil that is permanently frozen. And we know also that in this soil, the permafrost soil, there are in the Arctic 400 gigatons of methane that is locked in this frozen Arctic tundra. The microbial community constitutes the key to this lock. So if it thaws and if this comes out, then it's going to be processed and we're going to have huge impact on the atmosphere and release of methane to the atmosphere, which is a much more potent climate gas than CO2. So also, whoops. Okay, just try it again. Okay, so this shows a cartoon of permafrost, which has it's a very solid rock environment. It has a lot of ice wedges inside. It has a few plants, little biomass of plants, but we have photosynthesis going on. The plant is respiring and the carbon is going out by the microbes that are decomposing the carbon. When this starts to melt, we get more plants growing, the change of the ecosystem and the environment, get more carbon respired, and then you get more loop of carbon that is released to the atmosphere. This gives a quite distinct change in the ecosystems, like you have at least in the Canadian Arctic, where you have trees. You'll have the ground is just disappearing, and you have what's called the drunken forest. And also, if you go into the lakes in the Arctic, you find suddenly it changed in the last couple of years that you have a lot of these methane bubbles that are popping up, and it's getting warmer, and the methane gas is coming up. This has happened for a long time. I used to live close to a Merrimack Lake, and every winter we went out skating on this, and it was really fun to find these bubbles with methane gas and to light it. Just don't tell anyone back home that you're doing this. but it's really fun in the dark winter evenings in Norway. So this is going on for long always, but we can really see really big areas now that is very influenced on this. This shows temperature logging from the soil in Svalbard. So it's this first is the surface, 1 meter, and you can see that in 1 meter during summertime you have temperature fluctuations from -5 till +18 degrees, so really big change in temperature. And then in the winter it's frozen. So the first upper meter is active during summer with midnight sun and high temperature, and then it's frozen all year in the winter time. If you go 1.5 meter and below, you can see that it's permanently frozen soil in all this. And how long will this stay this way? Because we can monitor from year to year that these levels are getting higher up. Or the permafrost is going deeper. So what we do when we have classes in Adventsal with students, we go out after the permafrost or the soil has started to melt, the snow is gone, and we put out chambers. We put a little bit of soil in the chambers. We give them different substrate, and we measure, and we see the rates. And it's in just a couple of hours we can see really high stimulation and high rates in this. And that's kind of fun to do microbial ecology in a place like this because there's no big trees and plants around. So it's really easy to see the active guys in the environment. Yeah, so we can easily measure the gases coming out. And of course, we're interested in the composition. We need to know who are they in these samples. How are they influenced in the different carbon and nitrogen cycling? What happens when we have this thawing, when it's not permanently frozen? And also if there are differences in the different soils, and that's something I will come back to later because of the work that has been done in Berkeley lab. So just I don't know if all of you are microbiologists, but the complexity of soil, ultimate challenge, it's really difficult because you have all this the compartment where microbes are into the different, they adhere to the soil and silty clay particles, and it's sometimes really difficult to get DNA, to get the organisms out of this. And we know that it's a high microbial diversity. We know that only very little of what we have there has been cultured. It's really difficult to please the soil microbial organisms, especially from the low temperature area. And we were also interested to look if this change in diversity, that the mechanism that works for high organisms, would they hold for microorganisms. So we did a study a long time ago where I wanted to see if there were differences in soils from Bergen and then from Tromsø, above the Arctic circles, and from Longyearbyen. I did a conventional try because I was hoping that there was low diversity, low resolution DNA fingerprinting from the different soils. It was just too complex, too difficult to resolve this even by doing several steps of this PCR DTG analysis. If I do the archaeal profile, we can see distinct community profiles. We can also get information on the organisms there, and what we could see was methanogens that were dominating in the Arctic soil. The bacteria population was quite impossible to resolve using this. So what we did was to extract DNA from the whole community and measure the time it took to reassociate, to find its homologic DNA, to give an estimate on the overall diversity in this. In these different soils. And then the number that we got, we referred that to E. coli, a standard E. coli genome. And then you could see the Svalbard soil was 8,000 till 9,000 completely different genomes in the gram of soil. In the Tromsø soil, it was a little bit less, and then in the Bergen soil that I knew was high and highly diverse. It was a little bit higher than, or the same as we found in the Arctic. So this was kind of surprising to me at that time. This is a long time ago, because I was challenged that it was such a high diversity in the Arctic soil, and I was very excited, and I was eager to try to communicate this out to the scientific community. And this was the response I got, that this was just a ridiculous work of the Bergen Group that we've been doing for the last couple of years, and we should try to find something else to do. So it was not accepted. This is a long time ago, but the wind is now blowing in this direction. This is more and more data has been adding that there is high diversity, and the mechanisms that work for higher organisms are not the same as for microorganisms. Anyway, I tried to go back into the Arctic. I went out because I was doing the work was done in Longyearbyen, where the settlement is in Svalbard. So 1,500 people live here on a permanent basis. I went to Ny-Ålesund, which is a more remote place, and I decided to do more detailed survey on these samples. So we went to Glacier retreat, where we had one soil site that was the soil that was less than 10 years old, because the glacier has been going back. And then it was a mature site far away from the glacier. We sampled a semi-desert, Knutsenheia, and also an island where there was a lot of bird droppings and a lot of birds nesting out here. And then we decided an even another place further away, where we can only access by boat, very close to Bird's Cliff, very high inputs of nitrogen, which was called the 14th July Glacier. And we also sampled from this analysis, from this soil. And then we did the modern 454 pyrosequencing data set, and tried to see if this was extreme, and if this was how the diversity was in these different soils. So sample size was around 33,000 till around 19,000. If we have the data that we got back, but we also need to clean the data, and we could see that we have clean data in blue and noisy data in red. So we really decreased the diversity when we cleaned away some of the sequences using the Pyronise pipeline developed by Chris Quince et al. So after cleaning, only 0.2 to 1.5 could not be assigned to phylum level. So at least we cleaned away a lot of silly things, and we could keep on to phylum level those sequences that we still had. And then we had 36 phyla that we could assign, and 126 groups assigned to the general level. Again, this shows just comparison of the different soils, so much the same. We can see the bottom of the graph is the proteobacteria, so it was kind of the same. We could see here some changes in the very new soil that was from the glacier. On this figure, I've just put those that were less than 2.5% as others, and you can see this is what we were enabled to classify at class level. When we did refraction analysis of this, we could see that even though we have increased the analysis or the number of sequences, we are not in saturated. We're still far below what we, in order to get to know the whole diversity. And also when we do the PCA analysis, we could see that we have some clustering going on. These three are matured soils and established soils, so this makes sense that they cluster together. This is the soil with higher impact of nitrogen, and this is then the very recent soil. And we could see, if we look into what are driving it in a different direction, there was a really big OTU on this, which was a cumulative trophic organism. That made also sense that was in that. I would like we looked into the the diversity of the denoised and looked into different models to see how they distributed, and we could see that if we used the log normal model, it was really constant, and it gave it distributed quite nice. And what we were interested in knowing was that how much have we actually, by this analysis, how much have we sequenced, and how much do we need to sequence to get to, at least say, we can never do 100%, but how much do we need to do to get to 90%? And this is, if we look at the normal distribution, we should be somewhere, we should be somewhere up here, meaning that this is the number of sequences we need to get there, and we are, at the moment, we are the line over there. So we are really, even though we have done quite a bit of sequences, we're really not into any, we're far away from what's really in the environment. So what we can conclude from this study, again, this support the high diversity in these Arctic soils. We found 36 phyla, 8 were found to predominant across all sites. Protobacteria dominant in all soils. I think that's probably what you find a lot of different studies now. No consistent rank order of the 7 predominant phyla. The remaining 28 with less abundant phyla constituted just 5% of the assigned phyla. And also the taxonomic classification, as I was telling, the better protobacteria was present in deglaciated soil, acidobacteria in more matured soil. At class level, we find high diversity in all soils, and it was a considerable overlap in the OTU observed. So what we think is that there is a common pool of Arctic species. Each community formed very quickly. We can actually see quickly establishment of the communities. And then we have the rearrangements of the environment. We are not sure because we again think the sample size is too small. So we really need to infer that between 5 and 50 gigabases of sequences will be required to capture 90% of the metagenome. So do we need this? I mean, this is data. Do we really need this? We think it's nice to have as a kind of an outer limit of what you're going to analyze when you do this, to know if you're just doing the tip of the iceberg or if you're really doing the substantial group of organisms there. So this can be kind of a guiding of the explorer, where we are really interested in the most diversity and least individual, the taxa or the bacterial counts. So also what our data suggests is that the rare biosphere is not that rare. It's just that we're not really analyzing, getting enough into the data set. So we have, in our analysis, we have maybe analyzed 30% of what's there. We have 70% to go, and how far is it to go to the terra incognita? This is just based on 16S, but if we also want to know the whole metagenomic, what is the story then? If we need a complete sampling of a metagenome, and if you take this is very rough, but if you take the 16S sample data that we have, multiply by mean genome size and divide by read length, this would be the sequences required for metagenome in Arctic soil sample. And this means that we have, if we stick within log normal, we need to do 3 times 10 to the 12 reads, like terabytes, to get there. So again, it's a lot of things we need to continue analysis, to do analysis on. Then you probably all know this, but this is, the reason, I knew Janet from a long time ago, but I was so excited by the paper that was published in Nature in October, where they did this elegant, because all the data I present now has been done on the active layer in the Arctic. But she went out and her lab and got a permafrost sample, and this permafrost was thawed in helium atmosphere and then incubated and measured for the methane emission, and then it was kind of striking to see that just after 2 days it was a real burst of the microbial community in this sample. And that's also something when we see this big fluctuation, and from year to year I can also see the change in the soil structure up there. So an elegant publication that came, and based on the methanogenic approaches, you were able to define the different processes on the methane methanogenesis and methane oxidation, etc., all based on kind of putting this together. And also that you were so did such a great job to do a puzzling of this genome of an organism that we've never seen before, it's never been cultured before, and been able to put this all together, which is very exciting. And I think it really adds up on the data that we have from the Norwegian or European Arctic. But still, and I know, because I've been talking to some of the people in the lab, I know that this was a really hard work to do. It was very time-consuming and required lots of computer. But we have been analyzing also the sequence data that you've been doing. It represents a significant sequence effort to do this and to assemble this genome with just over 3.
Okay. Can you hear me now? Okay. So even though this was a huge sequencing effort, it's also really far away from what's really there. So it's so much in these samples that it's not analyzed yet, and really need to put a lot of effort in to get to know more of this. So I was in my title I was saying, I think it's such a big genetic potential in these environments, and we still are only analyzing the top of the iceberg. So it's genetic resources. We know it's high. We know it's really high activity. It's at very low temperatures. We need a polyphasic approach in order to do this. We can't only do the sequencing. We also need the cultivation, the omics approach, and etc. And I'll just briefly show some data that we have. Well, we have been using this Arctic soil as inoculum to look in wastewater treatments, and also we used wastewater as inoculum using different. Okay, sing star. So using different substrate, acetate, wastewater, and also put up at different temperature to see the effect on this. I will not go into details here, but just show you the species abundance pattern of this. Again, you can see the Arctic soil inoculum has a really large diversity in this, and shows much more rich than the wastewater inoculum. And also, when we look at the log normal predicted diversity, we can also see the acetate and then the Arctic soil inoculum giving the highest diversity in this, and also being very active. So what we also have been using is to look into wastewater treatment that is in reactors where it's usually you need high temperature to have these systems going. But if we also can use inoculum that could be go at lower temperature, we will be very energy efficient. And this shows then inoculum from the Arctic at different temperature, and you can see that the methanogenesis is going on really well at 4 degrees and compared to 15 degrees. I will end with a little. This is just because we've been talking of methane emission now, and if we look into the ocean, the marine system, the CO2 is very important to keep this system, the balance in the system. And we have been doing, colleagues of mine have been doing mesocosm experiments in this water in the Arctic, and see that it's really dependent on the community composition, the way that the CO2 has been turned around. And it was quite fascinating because it was set up an experiment in Denmark and one in the Arctic. And it was, everything was very consistent. And we could see that it was one species that was coming up in both of these experiments, the Thalassiosira, but you can see, and it's genetic almost identical. But you can see in the more temperate, it was long chain-forming diatoms. And the one that was put up in the low temperature is a small one. Even more? Okay, I'll try. Yes, I feel like a sing star. Okay, so again, these are it's the same organism, same in we've really studied this in detail on the genetics, but it's small, it doesn't form change, and it's really, really efficient in fixing CO2. So has a very high carbon fixation relative to nutrient uptake compared to the CNP value. So it's so many details in the organisms in this system that we really need to take into account. So kind of my take-home message to you is that the Arctic is intrinsically fascinating. I think it's also very important economically because we can use a lot of the information there to industrial processes and things like that. It controls gigatons of carbon that is locked in this tundra, and we really need to get to know more of it before it's too late. And also the diversity in this is quite controversial. So what I was showing to you in the beginning was the models and the prediction on the climate and temperature. This is now going in direction we really know that we need to get more information on the actual players in this system. We need to get more information on the microbes. And this we need to combine much more with the people working on the models and the mathematics around this. There have been already some attempt to try to incorporate the microbial data, biomass and enzymes and growth kinetics, which has been, I think it's really in the start. It could be improved much better, but that's quite an important issue on it. It's also important how we communicate this climate, change of climate, and we have in Bergen quite focused on with linguists and with the natural science to communicate this in a good way, so it's not just threat and drama. It really needs to be understood. This knowledge also needs to be consumed by the politicians and policymakers to take this seriously. And then probably most microbiologists here, but I just would like to end with this quote from a good friend and colleague of mine. He says, "I make no apologies for putting microorganisms on a pedestal above all other living things. For if the last blue whale shocked to death on the last panda, it would be disastrous, but not the end of the world. But if we accidentally poison the last two species of ammonia oxidizers, that would be another matter. It could be happening now, and we wouldn't even know." And that's my take-home message, because it might just disappear before we know what's there. So we really need to continue focusing on the Arctic. So with that, I'd like to thank my colleagues, collaborators, also Bill Sloan, Steve Colson, Tom Curtis, Jessica Green, Anders Lansen, my PhD students, Chris Krins, technician Eria Sundan, students Andreas Ausnes, and Janet's lab for hosting me. And I also have to acknowledge the Bjerknes Center, they gave me these models. We have this was a center of excellency that was established in Norway to look at the climate change, and they've done so well, and they've been now established as a permanent center with governmental funding. So I think they're doing really good work on this, focusing on the climate change. So with that, I thank you for your attention. And if there are any questions, I'm happy to answer. I haven't gone into detail. The data sets are there, so what I was presenting was actually the major OTU that was dominant to see why is it dragging in the different directions. What we can see is that if you have a little bit of impact on a soil, then you'll see a dominance of one. So if you do agricultural practice, for example, on a soil, you can see a big change in the overall diversity, and then you can see a change in some certain groups in that. I haven't looked into detail enough on this more general analysis to see if that is true. It also depends really where you put your limit, where you decide to look at your sequences, or if you go. The longer you go into define species, the more changes you'll see. So it really depends where you put the. And for this very broad analysis, I've put the level quite high, so to make a broader story.
So I hadn't seen your calculation for that you would need 3 terabases or terabytes, the same thing. But you know what's really interesting is it's the same approximate number that Titus Brown has come with completely independently,
okay,
and based on assembly of other kinds of data sets. So how much do you think this is dependent on the sequencing read links? Because I know that when we were doing when Sanger sequencing was being done, the estimates were much lower. Mm-hmm. Do you think that this amount of sequencing is due to the very short read lengths that we have now?
Yeah, I think the short read lengths will influence on the old, but yeah, somewhere. But still, this is very rough, and I think it's an underestimate. Still.
It's funny, it's the same.
Yeah. Yeah.
I have this biodiversity question. Part of your work you were talking about has been used in thinking about what controls the diversity of organisms in any particular environment. And for plant species diversity, I understand what their environment is, at least, between tropics and temperate and Arctic. And tropic is considered kind of a more stable environment with less variability. The temperate zone has a lot of seasonal, interannual variability in climate. The Arctic at some level looks less variable, but I actually wondered, just when you're thinking about microbes, how do you characterize the environment around your small samples? It's such a small sampling zone. And so what is it that you're thinking about as being their environment when you think about what controls diversity of this organism, this group of organisms across the planet?
Oh, this is a very difficult question, because I remember when I started to work in soil, I couldn't believe that it was enough space for all these organisms in 1 gram. So my professor, he said, well, if you think of people And you have to walk like 3 hours that direction to meet the next one. So you think it's crowded? No, probably not. So, and it depends on how you sample, of course, and what is the big sample. And that's why I also was showing the soil aggregates, because you have air pockets, you have water, and you have clay and mineral, etc. So it's really different. And then you have plants, you have plant room, you have exudates, so that's also an environment there. We have been thinking, why do we have this high diversity in these soils? And we have been thinking, are they because there are maybe not many grazers? Are they because maybe the viruses are not enjoying themselves so well there? But still we have, the more we are investigating, the more grazers are found and viruses have been found. So last year when we did the summer course, it was a person just looking into viruses, and we could find lots of viruses. So, things that I could say a couple of years ago, I would predict like this and that, and this is the environment, and I don't know anymore, because these barriers are not very clear in these environments.
So this is a two-tiered question. One of it is this glacier retrieving data that you showed. It was very interesting to see like the immediate response and then what happens to the soils when they get more mature and the glacier is not there anymore. So, and you didn't go much in detail. Would you like to comment on that a bit? What is your thoughts about why it's happening in the way that it is? Like, just certain species are more abundant in younger samples than the old. What is your take? And maybe.
For the glacier retreat, it was quite obvious. It was chemolithotrophic organism that dominated, and then you could see, and there was logic it was most stones and rock. It was hardly no humic acids there at all. So you need to have someone to start to build up this community. And then it's also, we have some data with the transect, and we can see gradually maturation of the soil. And the only way I can say it is because you go from a more lithotrophic primary succession of the community. No plants there, it's just rocks and so on, and it hasn't been exposed, only been exposed for 10 years. But still, we could see some of the same soil organisms.
So it's very interesting that you said that, because that was my second question. Because when we look at also in our soils, especially into the active layer where we have carbon, we see quite a lot of similarity to any other soil that we study with. Like, when one thinks about extreme Arctic conditions, one might think of we will find more specialized, understudied phyla in there, which sometimes is and sometimes isn't the situation. What is your take on that?
I think we need some time for that one. I don't know what to answer. It's not an easy answer to that, but you see, actually we did apply for a new project to go into detail to see the mechanisms, just do it much more with that as a focus, and it was rejected. Yeah, so I'm happy to discuss with you.
I really like the results on the diversity, that showing that the soil you're studying are more diverse. And I was surprised because, so I work with soil, a prairie soil, which is very diverse. But I thought that I would be less diverse, and that's why Rachel Macaprank could assemble a whole genome, because, as you say, with the shallow sequencing, as you explained it really well here, she was able to assemble the whole genome because it was not so diverse. So on one hand, I mean, your results are really interesting. And on the other hand, is that, it would be wastewater, for example, as you show here, really less diverse than something else, or do you have any other paper that, comparison, I'm sure you thought of that way more than I did.
I didn't want to say, but I think you had really good luck when you assembled your genome. The way it's probably, again, that was permafrost, and maybe that's probably not us, because that's a different story than the active layer. So I've been talking about the active layer, but this is very, again, it was, I think what you did in this, you have a substrate, and because I have been doing, a long time back, I tried to sterilize my soil, and I added substrate like methane, and then I had one that I flushed with nitrogen, and even though the diversity is really still, it's still high diversity, you have the evenness so different, so you get into one really bug that is so good, and that will grow, and you'll have so many copies, and that's why you manage to analyze it on top of on the rest. So that's why I think you managed to get this from that one, and it's also a quite small genome compared to what's we thought as a soil genome. When it comes to, well, I don't know, why do I think that they are so diverse still? I just think it. I wanted so badly to go to a place where it was easy to work on soil microbial diversity, and it doesn't work. Yeah, so you have to go into really sewage or something to find a much more simple community. And also, that's also why we have done all the enrichments to try to see if that would, what kind of population will it stimulate to go into different directions.
I actually think our time is up anyhow. So I'd like to thank Lisa for a very interesting seminar. Thank you very much for coming.